Hey guys, welcome back. You're doing a great job at navigating this semester. This week's set of material goes a little bit along with last week. So I introduced to you guys how the cell works, all the different parts of the cell, what those parts look like. And this week we're going to look at how those play together to actually do something for the whole entire human body. So as we look at how this is going to be done, I'm going to give this to you in a couple of different ways. I know everybody learns a little bit differently, but I'm going to first just kind of introduce why in the world this conversation is so important. And then I'm going to talk about kind of how this is going to be done. So first things first, the key concept for today is mitosis. Now, many of you have probably looked at this process in the past at some point. Potentially you haven't, but I'm going to give you some basics here today on how this works. So mitosis is a process of what we would call cell division. And cell division basically is being done here for a number of reasons. Those include things like growth of a cell, growth of the body, human repair. So, for example, one of the best things I could tell you about is something like your skin cells. And your skin is a set of cells that are always undergoing mitosis. They're constantly dying off. And mitosis basically means I'm going to take one cell and I'm going to create two identical ones from it. So, the key thing to recognize with mitosis is that we're not looking at reproduction of new individuals. We're not looking at any type of like metamorphosis. All we're doing is taking one cell and making two cells from it that are exactly like what we call the parent cell. Now, this once again is super, super important because many of your body systems work utilizing this situation. So if you didn't have the ability to undergo mitosis, your skin would never regrow. Now, think about your life as a child or if you're prone to accidents, okay, and you've scratched your body all up, okay, that would never heal. Basically, is what your life would look like without mitosis. If you never had mitosis, you would never grow from an infant. Okay, you would always stay that size. Mitosis means I can take cells, multiply them, and allow for growth and potentially repair to occur. Now, in this conversation, before I get to how it actually works, I need to mention to you why in the world we have it. So once again, it's a regulatory process that tells our bodies, hey, we can regenerate, hey, we can repair, hey, we can help ourselves out and live a little bit longer. But guys, if this process by chance grows out of control, there are certain checkpoints, which I don't talk about in this class. It's super complicated. But the reality is if you didn't have these checkpoints, what you would end up getting is something like cancer. And cancer for us is going to be defined as out of control cell growth. Okay, so if you know someone that has cancer or is undergoing some type of situation of a tumor or cancer that potentially has metastasized, we'll discuss all of these examples next week. But today, I just want you to know that that means mitosis is out of control. So you're supposed to get these signals that tell your body, hey, we've made two from one, we're good, let's stop there. Okay, we've got it. Without those checkpoints, what happens is those cells continue to grow and grow and grow and grow. And as they do, what happens is you get this accumulation of cells, which for us would mean that you have a tumor, okay, or in this case, cancer. So today's kind of conversation, I'm going to give you a video to watch that's a little bit more exemplatory as far as giving you a graphics. So I'm going to give you a really short version of this process, and then I want you to watch that video as well. That video will be more informative. And then later today, you're going to go through and fill out a worksheet that basically exemplifies for me, hey, I understand what mitosis and the process of mitosis will mean. So first things first, we have the process of mitosis. It means I have cell division and I'm going to have one cell becoming two cells, okay? And these cells here are going to be completely identical. Keyword, completely identical. One thing you forget about, or students will tell me about down the line, is they're like, yeah, these guys aren't necessarily identical. They are. They're the exact same copy as the parent, okay? Now, in this process of mitosis, that is highly regulated. And we begin with our first phase called interphase. Now, interphase is going to be our growth phase. If you look here with our growth phase, we're basically talking about how we're preparing the cell for division. In this case, the cell there, you can see on the outside, the nucleus in the middle, what's happening is that everything is multiplying. So think about it this way. If you have one cell and you're going to make two from it, if you don't multiply everything in that original cell, you're only going to get halvesies, okay? You don't want halvesies. You want the exact same thing in those cells. So I have to take my one cell and multiply everything that's in there by two so that when I split it, I have the exact same thing in both cells. So in interphase, there's some key things that are happening for us. Number one is that I'm synthesizing and replicating DNA. Remember, DNA is in your nucleus, and that DNA is the code that you become everything from. So all of your cell metabolism, all of who you are is coming from that code. So we're going to multiply and replicate that DNA in interphase, which is the very beginning preparatory stage of my process of cell division. You're also going to multiply organelles. These organelles, like we studied last week, all of those have to be multiplied so that when I split this cell, once again, I have an even amount in both of them. Otherwise, I get half in one and not so much in the other, and things don't work the way they're supposed to. Lastly, you also multiply all of your cytoplasm. Now, cytoplasm, we know, is that jelly-like substance on the inside. That jelly-like substance is going to be kind of the holding tank, so to speak, of everything that's in there. All right, so our first stage of actual mitosis itself, so interphase is preparatory. That's our pre-phase, you could say. Now we're going to go into 4 stages that are actually dealing with the process of mitosis. And mitosis, by definition, means I'm dividing my nucleus. Okay, so that's what we're worried about here. We're trying to figure out, how do I take this DNA and divide it in half? First stage is going to be prophase. Prophase has 2 different things happening for it. You see that nucleus inside of there. You see the cell. In the case of prophase, I would give you one main thing that's super important, and that is that the chromosomes are being made. So what you have before prophase is a bunch of just loose DNA. That DNA is hanging out in the nucleus. It's just kind of like stringing around. But when you come to prophase, all that chromatin, all of that loose DNA is going to condense itself up and become what we call chromosomes. And these chromosomes are things that I can actually use to divide then. If they're not condensed into chromosomes, I can't divide those cells out. So I have to do that first. Then what we have is our next stage called metaphase. And in metaphase, you can see here how I have my chromosomes lining up along that metaphase plate. All those chromosomes that I just made in prophase are now lining up, okay? They have to go through this process of lining up, because in a minute I'm going to split those guys. Now, I want to bring your attention to one of these things, okay? This chromosome is an X. Y'all see the X? Each side of that X, okay? So if you have an X, each side of it, this side, each of those are called a sister chromatid, okay? Now, the sisters are going to get split, okay? So this half is going to go to this part of the cell, and this half is going to go to this part of the cell. That's the whole idea of division. So when you look at that, you're going to have to tell me about that process later. But when you see that, metaphase just means, hey, I've lined these guys up on the middle of that metaphase plate, or right down the middle of the cell, and each of those X's are going to split. They're called sister chromatids. So when I begin to split them, we move into our next stage called anaphase. And anaphase, you can see here how they've split and they're being pulled to one side. So each side here is pulling a set of chromosomes or sister chromatids to each side. That's telling us that, hey, we're in anaphase. Now, anaphase is important because what's happening here is that I've now split my chromosomes apart. My sister chromatids are pulling to each pole. Each of those poles then are going to take part of my DNA. Okay, and in a minute, we're going to create new cells from that. So the whole objective of anaphase is to split the actual chromosomes. You can see they do that by a very special guy called spindle fibers here. These spindle fibers are kind of like, they're really small. I think of them a little bit like cobwebs or something. They're just really, really thin, and they just pull. What they do is they attach to those centromere of the actual chromosome, and they pull apart. Next, we got telophase. Telophase is one of my favorite stages, because what happens in telophase is you get the 2 nuclear envelopes that reform. So that basically means, hey, I have my DNA in here now. Okay, I pulled each of those from each side. And when you have that DNA, basically it has to reform its nucleus around there. So both of these guys are reforming in telophase, and then something else is happening here. And that's what we call the cleavage furrow. Now, and that's not inappropriate, but this is called a cleavage furrow for obvious reasons. Okay, this cleavage that you're seeing here, this separation, is basically going to be where a constriction happens, and I end up getting 2 completely different cells pulled apart. Okay, and so that cleavage furrow happens in telophase. Super important for us to know that. Lastly, I got 2 identical cells, just like my parent, same size. Okay, I've got these 2 new guys. What they're going to do then is they could potentially undergo interphase again and go back through that process, multiply, create new cells. That's fine. But their main job is over. And so those 2 main cells are going to go do their do their work, whether they're a skin cell, liver cell, cartilage cell, whatever, and go take care of the business that they were asked to take care of. So once again, this is the main process of mitosis and how it works. You will watch a video on this as well, answer those worksheet questions that I have for you, submit those back to me. And then next week, I've got some really unique kind of assignments when it comes to cancer, basically because that's a relatable topic to everybody. So hope you guys are having a great semester, and email me if you have any questions. Overall, excellent job. We'll see you guys soon.